Hello, good morning. Myself, I am BSR Botany lecturer. Today our topic transcription. Transcription means synthesis of mRNA from the DNA in presence of RNA polymerase. In presence of RNA polymerase is called transcription. DNA dependent RNA synthesis, that is RNA polymerase. Not only mRNA, rRNA, tRNA, all are synthesized from the DNA. Important point. In prokaryotes, now one more important point. In prokaryotes, only one type of RNA polymerase is present. Only one type. Whereas in eukaryotes, three types of RNA polymerases are present. RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. RNA polymerase 1 is involved in the synthesis of rRNA. RNA polymerase 2 synthesis in the synthesis of mRNA. And RNA polymerase 3 is involved in the synthesis of tRNA. RNA polymerase 1, rRNA, RNA polymerase 2, mRNA, RNA polymerase 3, tRNA can synthesize. Especially in RNA polymerase, two parts are present. One is core part and another one is non-core part. In the non-core part, mainly two subcomponents are present. That is sigma factor and rho factor. Sigma and rho factor. Now we are going to transcription process. You just look at there, in the DNA, two strands are present. You know the two strands. In this two strands, one strand starts with 3 to 5 prime. This is 3 to 5 prime. And this one is anti-parallel. So this is 5 prime to 3 prime. Especially during the transcription, you know RNA polymerase enzyme. Along with RNA polymerase, promoter and terminators are involving. Promoter, here the promoter is attached to the two strands. One at the third end of the one strand, fifth end of another strand. This is a promoter. Promoter containing special nucleotide sequence. So that is why it is a promoter. That zone is called Tata zone. Remember. And terminator. So this is the terminator. Terminator is also containing nucleotide sequence, which terminates the mRNA synthesis. Terminates the mRNA synthesis. So especially in the RNA polymerase, you know RNA polymerase enzyme involving in the transcription. This RNA polymerase enzyme, main two components, the sigma factor and rho factor. Sigma and rho factors are involving. Mainly the sigma factor, initiates the mRNA synthesis. So now the mRNA synthesis initiated by sigma factor of RNA polymerase. Then RNA polymerase is moving on DNA strand like this, in this way. So that is why mRNA can be synthesized. This is DNA sequence. Suppose here thymine, adenine, cytosine, guanine, adenine, thymine is present. So what type of nucleotide can be seen in the mRNA? C complementary with adenine, A complementary with thymine, but here uracil comes. If there is C, here G comes. If G is there, C, here G, C. If A is there, what is that one? Here thymine should come, but uracil, like that adenine. So then third and to 5 prime is the template strand. So this is the template strand. This is the template. Yeah, third and to 5 end. That strand acts as template. when that act as template, this one is called as coding strand. This is coding strand. Coding strand nucleotide sequence, mRNA nucleotide sequence same. Coding strand sequence, mRNA nucleotide sequence is same because these two strands are anti-parallel to template. So that template is involving in the synthesis of mRNA. This is mRNA. So the RNA polymerase is moving on DNA strand. mRNA is synthesizing there mainly in the terminator region. The rho factor stops the RNA synthesis, which stops the RNA synthesis. So when it stops the mRNA synthesis, that mRNA can release into the cytoplasm. mRNA can be released into the cytoplasm. There mRNA involved in protein synthesis. mRNA involved in protein synthesis. Whereas in prokaryote, both transcription and translation takes place within the same cell. It means there is no nucleoplasm and cytoplasm difference. But in eukaryotes, transcription takes place in nucleoplasm, translation takes place in cytoplasm. In prokaryote, entire takes place in the cell. Because there is no differentiation like nucleus and the cytoplasm. So this is the transcription. Thank you to all.